Hello and welcome to Nikolai's genetics lessons, and today's problem is the following pedigree described in 1937 by Beard shows the inheritance of X-linked color blindness and hemophilia in a family. What is the genotype of Tutu? Do any of her children provide evidence for recombination between the genes for color blindness and hemophilia? Take a look. We see here in the first generation male who has hemophilia and phenotypically normal female. And this couple has two children, and their son has color blindness. The daughter seems phenotypically normal. She marries another man unrelated to this family, and this couple has three sons. One has hemophilia and two has color blindness. So we have to find why this happened, and is it normal inheritance or it is due to recombination. So take a look. Both these genetic disorders caused by certain mutations on the X chromosomes. So different genes, but both these genes we can find on the X chromosome. And for example, genotype of a male in the generation 1 would be so this is X chromosome, this is Y chromosome. Y chromosome is much smaller than X chromosome, and this man has a genetic disorder, so I would put this dot here. So we would know that this genetic disorder in one of the genes would cause this condition when person, if he would have a cut, his blood would run and doesn't stop. This is what happens usually when person has this genetic disorder. We see that the son has color blindness, and we know that he have to get this defective gene on the X chromosome from his mother side. One of the X chromosomes, so mother has two X chromosomes, and one of this X chromosome has this defective gene. But mother's phenotype is normal because she has one normal X chromosome with normal gene on it that produce normal protein. So phenotype of the mother is normal. So let's find out what happened here in the second generation. Let's build a simple Punnett square. So we take these two people, male on the top, so he has X chromosome and Y chromosome, and female here on the side, two X chromosomes. And now let's build simple Punnett square, and let's see what are possible genotypes in their children. Let's also add defective genes here. This dot would represent defective gene, and this red dot also would represent defective gene. And these genes have to be in different locations, of course, on these chromosomes. What we have here, one X chromosome inherited from the mother side with defective gene, another X chromosome inherited from the father side with another defective gene on it, normal X chromosome inherited from the mother side here, and defective X chromosome inherited from the father side here. Here we see defective X chromosome inherited from the mother side, and normal Y chromosome from the father side. Normal X chromosome inherited from the mother side, and normal Y chromosome inherited from the father side. So again, this is father side, this is going to be mother side. 50% of the progeny are going to be females, and none of the females are going to be affected. Take a look. This female are not going to be hemophiliac, because she has another chromosome inherited, X chromosome inherited from the mother side, which has normal gene. And one defective gene is not enough for female to manifest this genetic disorder. Let's take a look at this female. She inherited one X chromosome from the father side with defective gene for blood clotting factor, and another X chromosome which also has a defect in one of the genes which cause color blindness. But again, this is recessive genetic disorders, and this female has one normal gene for color blindness and one normal gene for blood clutter. So she is a carrier of both these genetic disorders, but she doesn't manifest these genetic disorders. Her phenotype is normal. And as for the males, here 50% of the progeny, of course, going to be males. Males has one X and one Y chromosome. We see the chances that this couple would have male or son with genetic disorder would be 50%. And this genetic disorder going to be color blindness, which this male would inherit from his mother side with X chromosome. From his father side, he only can inherit Y chromosome. And as for this person, this is going to be male, but from the mother side, he would inherit normal X chromosome, normal Y chromosome from the father side. So this family would have 100% daughters who are going to be phenotypically normal, and 50% chances that the son would be affected. This is what we see here. So we can put these genotypes here. So genotype of this person would be defective X chromosome inherited from the mother side, and normal Y chromosome inherited from the father side. And as for this genotype of this person, two variants are possible. Let's take a look. Let's assume that genotype of this male is normal. So he has normal X chromosome and normal Y chromosome. We know that all these males inherited Y chromosome from their father. That's what makes them male. And they got X chromosome from their mother. Mother have to have genotype which is shown here. In order to explain what we see here, this female have to inherit this defective X chromosome from the father side and this defective X chromosome from the mother side. Take a look. One of her X chromosomes has a defective gene for hemophilia, and another X chromosome has a defect on one of the genes that cause color blindness. Color blindness and hemophilia. So we know that color blindness would be inherited by these two sons from their mother. They have to get this defective X chromosome with defective genes on it, and another son got another X chromosome from his mother, which cause hemophilia. This is X chromosome, and here defective gene on it. We can explain what we see in this pedigree without any recombination. So no recombination happens here, and genotype of this female would be double heterozygous. She's going to be heterozygous for blood clutter factor and for color blindness. So her phenotype would be normal, but 50% of her sons may be hemophiliac, and another 50% may have color blindness. We can say that recombination would happen here if some of your sons would have both color blindness and hemophilia. But because we do not see such combinations here, we can say that what we see here can be explained with just normal segregation of traits and no recombination happen here. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day, and see you in the next video. Goodbye.